Good afternoon, everybody. Thank you, Molly, for this fantastic meeting. It's just amazing to see all these very interesting talks. Okay. And I must say that it's like I'm a jazz musician, and I feel like I am in a concert by Herbie Hancock and Wayne Shorter, and I am asked to play during the jam session, and it's just amazing to me. So I'm going to try to give an interesting talk. So actually, I'm not the lead author of this study. The main author is John Welsh and Alexis Simon, together with Hilde Schneemann and Christel Freys. So you have their picture here. And believe it or not, they are all four in this room today, so I can acknowledge them directly. Okay. So hybridization is very widespread in nature, and also it is used by humans. You know that maize, for example, are hybrids between inbred lines, or these cherry tomatoes. Actually, this is the strain I am using in my organic garden. It is very robust, and the tomatoes are very sweet. My children love these tomatoes. I don't know why usually people doing organic gardens are against hybrids. So there's no problem with being hybrid. It can be used to make new varieties. For example, this pine berry, a strawberry with a taste of pineapple, is a hybrid between a South American and a North American strawberry. You have this dog, Pomsky. I discovered this strain because of my daughter. She wants a dog, and she finds it so cute. And actually, it is a hybrid between this breed and this breed, a husky, a well-known husky, and this one is a dwarf Spitz. It's also true for beer. So I don't know if you like beer, but maybe tonight, if we are lager together, the yeast used to make a lager is a hybrid. It took us some time to find the other parent. One is Cerevisiae, the other is Eubayanus. And now they are trying to hybrid them again to make new beer. It's important in health and medicine for virus. You know, they can recombine and become more virulent. It's even an issue in cancer, where you can have cellular fusion in leukemia or mitochondrial capture. It's important in conservation biology. So in conservation biology, it's complex because sometimes it's good. It's called genetic rescue. So it helps to rescue a population. And sometimes it's bad. It's called gene pollution or extinction by hybridization. And I must say it's not very clear to me what is the difference between the two processes, except what people think is a species or not. So you have many examples of hybrid zones, and it's what I am doing for 20 years now. I study a hybrid zone between two mussel species, and I put two other hybrid zones that I like a lot between toads and in snapdragons. And you all know that now we know that human has a reticulated evolution with hybridization between Neanderthal and Sapiens, or Denisovan and Sapiens. And it has helped adaptation to altitude in Tibetans with the Denisovan allele as these genes, EPAS1. And actually, Tibetan dogs also adapted to high altitude thanks to wolf allele at the same gene, EPAS1. Okay, what happened? Sorry. Jenny? Yeah. I think I need your finger. Yeah. Yes. Okay. So yes, I put hybrids under every bush because I use this quote given by Wagner and quoted in Abbott et al. Wagner said, "We used to make fun of Edgar Anderson by saying that he was finding hybrids under every bush. Then we realized that even the bushes were hybrids." And Anderson published his book about hybridization in 1949, 70 years ago. And still now, people sometimes make joke of people seeing hybrids under every bush. And every year, for 70 years, we have a paper saying that, oh, we thought hybridization were rare, but now we think it's more frequent. The problem with hybridization is that we don't really know what is a hybrid. It's the same debate as for species. As we are unable to define species well, we are also unable to define hybrids well. And at least we should consider divergence when we talk about hybridization. So speciation is a gradual mechanism that proceeds through intermediate stages during which hybridization is not completely interrupted and genomes remain permeable to genetic exchange. So it's well known in Drosophila, this paper by Kohn and Orr, that the success of hybridization between pairs of Drosophila species decreases with divergence. I reverse the figure, usually it's in the other direction. But I reverse it to compare with this paper by Camille Roux. Actually, Camille is also in the room. Hi, Camille. And here, the author used demographic reconstruction to identify gene flow between populations or species. And they identify pairs of population or species with homogeneous gene flow, a pair of population or species with no gene flow, and pairs of populations or species with heterogeneous gene flow, some region of the genome with gene flow, and some region of the genome without gene flow, defining a gray zone of intermediate stage between one species and two species, a stage with semi-isolated species. So to explain the fitness of hybrids, we usually have two models in the two extreme cases. When the line or the population are very close, we usually observe hybrid vigor, and it is well explained by Wright's single locus theory. So it's actually deleterious mutation. You have accumulation of deleterious mutation in line 1 at the homozygous states, deleterious mutation in line 2 at homozygous states. And if you do the F1, all the deleterious mutations are heterozygotes. If you do the backcross, half of the mutations are heterozygous. So you expect heterozygosity to explain the fitness of hybrids. In the other side of the spectrum, you have hybrid breakdown. The hybrid is less fit than the parent, and it is well explained by the Dobzhansky-Muller incompatibilities. Claudia yesterday reminded us what is Dobzhansky-Muller incompatibilities. You have the genotype at two loci in species 1, in species 2, and one allele, big B at locus 2, is incompatible with big A allele at locus 1. And when you have this genotype in the hybrid, it is counter-selected, while the other combination, that is often the ancestral state, is perfectly fit. So we now have many examples of molecular mechanisms that explain hybrid breakdown. So, and actually, all the talk yesterday, I thought maybe it's again a new way to make hybrid breakdowns with a proliferation of satellite motifs very quickly, or new spectrum of mutation, but you have many other mechanisms. But if you think it a little, all these mechanisms, first they highlight that there is many ways through which immensely large and junky genomes can become incompatible because of selfish elements, intragenomic conflicts, runaway process. But also, they all retain the spirit of the Dobzhansky-Muller model. And if I just take the model of translocation by resolution of gene duplication, it's exactly a Dobzhansky-Muller incompatibility. In species 1, where you have a null allele at locus 1 and the gene present at locus 2, the reverse for species 2, and when you do a recombination, you can have a genome without the gene. and so the hybrid is dead. So following the discussion this morning with the infinitesimal model and the omnigenic view of adaptation and phenotypic traits, complex traits, here with hybridization, we don't need really a complex model because we can kill hybrid very easily with this simple determinism. And it's what I thought for a long time. And now I changed my mind. And it's what I'm going to try to explain, if I'm not too slow. So first, the beginning is that I am studying hybridization in the middle of the continuum in muscles and in Sayona C-squarts. So they are here in the graph at the end of the gray zone, and even C-squarts, they are too diverge. It's difficult to identify gene flow between the genome, but they can still hybridize. In muscles, this is a complex of several species with mosaic hybrid zone. But what I want to say today is that when we try to cross muscles to find the Dobzhansky-Müller incompatibilities, we failed to find some. First, we found no departure from Mendelian expectations, and we only found one strong DMI, this one, but the net effect was heterosis. The double heterozygotes was the fetus genotype. What we found is rather an excess of multilocus heterozygosity. Here you have the hybrid index of the offspring, here the heterozygosity, and you have an excess of hybrids with higher heterozygosity than the expected half. And in muscle, we also have admixture in ports. It's due to a human introduction in several ports in France. They are found only in the port and not outside of the port. And they are strongly admixed, like a hybrid swarm, some people would say, or maybe a hybrid species, I don't know. But what is surprising is that first, it's the first time that we observe this intermediate admixture in muscle. We never observed this intermediate admixture in muscle before. And when we took all the loci that are nearly diagnostic between the two species, so it's only a few SNPs in the genome, all the SNPs are equally intermediate. We don't see one SNP that is Galloprincealis or the other that is Edulis. They are all really intermediate. And also the DMI found in the cross is also polymorphic in the port. In Sayona species, it's even worse. The divergence is 15%. Can you imagine that you can cross genomes that are this divergence? And actually, you can. And when you look at the segregation distortion and the F2, it's quite okay. Here you have the frequency of the heterozygotes and the two homozygotes. And first we found only a few departures from Mendelian expectation, and it's just heterozygote excess in this chromosome and this chromosome. But the major result is similar to muscle. It's an excess of multi-heterozygote genotypes. So here I rather see than this excess of heterozygotes, and maybe just by chance you can cross the significant threshold, but I'm not sure that you have really a DMI or something like that in the middle of these two chromosomes. So we needed a new model, an alternative to the Dobzhansky-Müller model. And the idea is to have a chain of substitutions from parent 1 to parent 2 according to a simple model of divergence. One population diverge independently from the other without gene flow. And you have a chain of substitution, mutation 1 giving phenotype 1, etc. You go through the common ancestor, and then you go to parent 2. And we use Fisher's geometric model following this very influential paper by Nick Barton in 2001. So it's a similar model. And so you have a fitness landscape. several traits. And mutations can be deleterious, so the same mutation can be deleterious. In another background, it can be neutral, and in another background, it can be deleterious. So the spirit of the Dobson stimuli is there in the model. You can have a mutation that's fixed because advantageous or neutral, and you put it in another background, and then it becomes deleterious. Okay. So to have our hybrids, you imagine the ancestor, you have a chain of mutations going to parent 1, and the position of the loci in the genome, a chain of mutation in parent 2. This is the haploid model. To have the hybrid, you just sample randomly the mutation, and you have the hybrid with a given hybrid index. We can develop a diploid model. In this case, we used additivity and the phenotype. So the parents are homozygotes with 2 mutations each time, and at the same time, to make the hybrid, you sample the mutation randomly to create the hybrid with a given hybrid index and a given heterozygosity. And we want to describe the fitness of hybrids with this simplified genotype, hybrid index, and heterozygosity. So again, I put the chain to explain the best I can. Here you have the phenotypic distance to the optimum in parent 1, in parent 2. You can start from parent 1, and you have all the mutation in the order of evolution, how it proceeds during the divergence. You go through the ancestor, but you do not really need the ancestor to predict the fitness of hybrids, because when you took the mutation randomly, you can have many paths, and it resembles a Brownian bridge interpolation. And so John has this wonderful idea to use this approximation to obtain the fitness of hybrids. I wanted to say that it's not only, to me, it's not only an approximation, it's also a way to have a new view of hybrid genotype. It's like retyping evolution in a random order. So you know mutation order speciation, so it's the same spirit. If you're creating an hybrid, is really to retype evolution by sampling the mutation at random, and it's no longer dependent on the fitness landscape. So this is the basics of the model. We have the breakdown score. It is a phenotypic distance to the optimum, the sum on all the traits. There is a scaling factor. You can define a fitness function, but here we give the result only on the phenotype, not because fitness is important for the true evolutionary path, but we want to demonstrate that it is not important for the phenotype of hybrids. We define for mathematical purpose the scaled breakdown score, which is the breakdown score divided by the worst hybrid. And to have the breakdown score of the worst hybrid, you use a standard Brownian motion that is not constrained. So maybe I should have said that what is a Brownian bridge? A Brownian bridge is a Brownian motion where the arrival of the walk is constrained to be at one position, while the standard Brownian walk, you can go in any direction. Okay. So we give the result as a function of the hybrid index, heterozygosity, as well as parent 1 maladaptation and parent 2 maladaptation. And it turned out that these two parameters are very important to predict the fitness of hybrids, and it's something I did not realize before to work with John on this model. And it's also the interest to develop a model is to change your mind, to have a new view. It's already the end. Stop. Okay. So briefly, we did some simulation, and the Brownian bridge approximation works very well. It works very well if evolution is nearly neutral around a fitness optimum, or if you have adaptive walks to a new optimum. As soon as you sample the mutation randomly, you have the same result. It's independent of the history of adaptation, and it works very well, the approximation. Okay. So we can find if the two parents are very far from the optimum, near the worst possible genotype, we find the right expectation. The fitness depends only on heterozygosity, with P1 being the worst genotype, and the F1, that is fully heterozygote, is the best genotype. But in true value, you often find that the F2 is a little less fit than the first backcross, while the heterozygosity is the same. So you just have to include a little bit of adaptation in the two parental lines, and then you obtain this curvature, and you fit very well to the data. When the two parents are well adapted, then you have this landscape. The parents are fit, the F1 are fit, and the worst hybrids are in this parameter space. And it fits well with the result we obtain in mussels and Sayona, with an excess of heterozygotes in the F2, but not in the backcross. And sometimes you can find an excess of heterozygotes in the backcross, and it can be explained by parental maladaptation. And it's what we observe in poplar tree, Populus, and this plant, Senecio. We have a little bit of parental maladaptation that give a curvature in the fitness landscape. Okay, so maybe I'm too slow, so I skip that. Oh no, I said, sorry. I wanted to say that one important question was that Fisher's model, it was said by Barton in 2001, Fisher's model does not generate the strong effects of single chromosomal region often observed in species crosses. And it's true with a standard Gaussian fitness function, but if you play with the curvature of the fitness function, you force epistasis, and it is this parameter k in the fitness function. You have a fitness function that is more like a table. In this case, you can have speciation genes. So genes that fixed with a low selection coefficient, exactly as we observe in molecular evolution, but with a strong selection effect when introduced in the other parental background, but not when with the standard Gaussian. So we were happy with this. We can do that, but I recently changed my mind, because if we look at the marginal fitness of these speciation genes in the F2, it is not correlated at all. It means that these speciation genes do not contribute more to hybrid breakdown in the F2 or backcross or intermediate hybrid. But in speciation, what we want is to kill hybrids. We don't want any hybrids. The fact that one chromosomal region is unfit in the other species genome, it's another question. So the name speciation gene is maybe not very accurate, and the true speciation genes are all the mutations that contribute to hybrid breakdown in the first generation hybrids. Okay. So to conclude, hybrid fitness is well explained by a simple polygenic model of stabilizing selection on many traits. The Brownian bridge approximation works well. Predictions are independent of the mode of divergence, nearly neutral or adaptive walk, and of the fitness function. And actually, these two results are the same, to my point of view. Just the Brownian bridge approximation works well because it's independent of the evolution during divergence. So this model and the data we fit to suggest many substitutions contribute to a species barrier, even when strong effect mutations are identified in introgression experience. To me, it became two independent questions: introgression experiment on one side, and to explain hybrid breakdown of the first generation hybrids. Parental fitness contributes to asymmetry, and Hilde and John are incorporating dominance now, phenotypic dominance in the model, and they are beautiful results. And this model, the Fisher geometric model, together with the Brownian bridge approximation, open new perspectives to analyze data at the genome scale, because we will have an alternative to map incompatibilities, to map barrier locus. We can reason at the scale of the whole genome and fit the data, whatever the causal genes, to the level of ancestry, the size of the chromosomal tract, and things like that. So it's very exciting. I thank a fellowship by the French Embassy in the UK and the Churchill College that allowed me to visit John's lab during 6 months in Cambridge last year. So it was a wonderful time. I met Chris Jiggins. I met Prof. Edwards. Prof. Edwards is the last student of Fisher. He's the Edwards of Edwards and Cavalli Sforza. So it was fantastic to meet him. And we also have good beer. I don't know if the yeast is hybrid in the UK. And I thank you for your attention.